right, so I could explain a little bit what happened there. So Ed Boyden had to cancel on a very, very short notice. And since I was scheduled to be here anyway, I promised Zhang that I would step in and give another talk instead. And given the short notice, I thought when Zhang asked me that, well, no problem, I gave a talk to high school students in Sweden in the spring, so I have a lecture all ready to go. And then when I arrived here yesterday, I realized that lecture is in Swedish. And I also realized yesterday that in the audience there were also students from HKUST and not only high school students. So what I will offer you today is kind of maybe a mixture of something that will be nice to hear for high school students, but also something that may be on a college level or so. But essentially it's all comes down to very basic chemistry, basic chemical questions, but are quite central to biology in a way. So the title is here, Induced Fit, and I will tell you a little bit about how we can study ligand binding in proteins and how we use nuclear magnetic resonance to do that. So this is kind of a research talk with hopefully a little bit more pedagogical aspects to it. So here, what you see here is a beautiful representation of a cut through an E. coli cell. This is a drawing by David Godsell. And what you see is a big mess. But these are basically macromolecules, proteins and RNA, DNA inside the cell. And it's a pretty thick soup in the cell. So obviously, all these molecules interact with one another all the time, and the proteins will bind ligands, that is to say, other proteins or small molecules or other biomolecules. And this is key then, this binding, the molecular recognition between molecules. And here's another Dave Godsell representation of a antigen-presenting cell interacting with a T cell. There's a red dot in the center. See if I can, there we go, here. And this is a small peptide from a virus that is bound to the MHC proteins on the surface of the antigen-presenting cell. And so the peptide has to be bound in a very specific way to the MHC protein. And then it's recognized by a T cell receptor on the surface of the T cell. So these delicate aspects of ligand binding is really totally critical for biological function. This is what basically kicks off the immune response. And when a protein recognizes, binds to a ligand, it usually has to change its shape. So what you see here, for example, is a 4-helix bundle protein, helices in red and yellow, and a short beta sheet up here. That's kind of closed, right? And then the protein can take on another shape where the helices move out, and underneath it presents a surface. In schematic view, here you have the closed form, the open form, and now that it's open, it can bind the ligand. And you can think about proteins as small molecular switches that turn on or off cell signaling in and between cells. So on, off, etc. Now, this is exactly what we want to study. How does a protein bind a ligand, and how can we study this? And that's what I hope to tell you today. So you can think about the process of ligand binding kind of like this. And this is the so-called induced fit view of ligand binding. It's the classical textbook case How many of you in the audience of the students have heard about induced fit? Do you come across this in biochemistry? I see at least a few hands. That's nice. So this is what's basically been dominating the textbooks at maybe high school level, but certainly college level, from all the way back since the 1960s. Koshland introduced this term, I think. So what you see here then is the protein is closed. It cannot bind the ligand. The ligand approaches the protein and kind of induces a fit in the protein. So it makes the protein change its shape, and now the ligand can bind. But there's an alternative view, and this is sometimes called conformational selection, where the protein is spontaneously undergoing this conformational change. So part of the time it exists in this conformation, and then the ligand can come in and bind. And you see these are different, right? And in the last maybe 2 decades or so, maybe 3, more and more evidence has appeared showing that this is a very plausible mechanism for ligand binding. You may ask, why do we care about the difference between these? And one answer, there are actually many answers, but one answer could be that if it is the case that you have, say some disease, and the protein for some reason needs to be in this shape in order to activate some signaling process, you may devise a ligand that binds and locks it in this shape. But if you then realize that this one is already present, you can perhaps devise some other ligand that binds in a different place, here, say, and that doesn't block the site so that the protein can still function in a normal way, but you've shifted the population and the signaling strength, so to speak. So allosteric drug initiatives kind of can use the fact that this process may exist. And it has been maybe, I think, surprisingly a lot of controversy between these two views. People have been fighting, is it this or is it that? But I think you can all recognize that when you look at this, it's really this view that we should apply, right? That you have both the induced fit and you have the conformational selection pathways. Both of these can be present at the same time, obviously. And I've also here indicated that it's not a one-way arrow. These are equilibria, so the processes can go in both directions here. So how do we study this? Can we disentangle what's going on for a given protein, how it binds a ligand? And we think we can. And we think we can do more than that. There are other questions associated with this if you want to address the question, does it bind this way or that way? So for example, you can realize that the affinity for the ligand of this conformation and that conformation, any difference in these affinities will play a role in dictating which pathway is active. And you can also think about the question, does the ligand change the energy landscape of protein conformations? Now I realize that the concept of energy landscapes may be unfamiliar unless you were here yesterday and listened to Alan Firth's talk. So here I have almost exactly the same type of figure as Sir Alan. So this is basically, let me go back. What I show is an energy diagram going through this pathway. This pathway, I think it is. The upper, yes. So we have on the vertical axis, it's free energy, and the horizontal axis is the reaction coordinate that tracks how the process is moving forward, so to speak. And here we have a difference in free energy between the ground states, 1 and 3 in this case. This corresponds to the affinity that you may recognize, the equilibrium constant, given by the exponential of the delta G naught divided by RT. And we also introduced the concept of an activation barrier. This is the barrier going from 3 to 2 and climb over here, and this dictates the reaction rate constant in the same fashion, and then there's a prefactor here. So the question I asked was, what's happening to this energy landscape when the ligand is present compared to the case when the ligand is not there, and it's just the protein's conformational changes that we are looking at. So the protein that I will introduce as the model system for studying this is, well, it's an interesting protein in its own right. It's called galectin-3. It is involved in cell cycle regulation, differentiation, apoptosis, and it is an actively pursued drug target by several companies in the world. And we'll be looking at the ligand binding domain, and ligands are galactosides, and here is a very small version of such a molecule. It's lactose, milk sugar, very common, and this is like the smallest building block that represents the natural ligands of galactin. And here you see the protein in gray. This is a beta strand, so it's a beta sandwich, and in gray the backbone trace, and then we have lactose in green with its oxygens in red, and then in yellow the side chains of residues that coordinate, that kind of hold on to the ligand in the binding site. So the name of the game now is to try to sort out which way a ligand binds, is to basically measure all of the different reaction rate constants here. And when you look at it, I'm sure you realize we don't have to measure all of them because it's a cycle, so we can get away with measuring quite a few less. Also you can invoke a few approximations that I won't cover in this talk. So what we use, the method we use is nuclear magnetic resonance spectroscopy. And this is a method that uses the intrinsic properties of the atoms. Atoms have a quantum mechanical property called spin, and they behave like little bar magnets. And if we send in radio waves, we can basically make these little bar magnets get excited, and we can do spectroscopy on this. And I'll show some example spectra on the next few slides. The beauty with NMR is that you don't need to label, you don't need to put any tags on the protein or anything. The atoms themselves are like perfect little spies that just sits there silently and report back to you what's going on in the protein. It could be structure, interactions, what happens when the protein binds a ligand, and it could be to monitor how the protein moves in time. And that's, of course, monitoring movement is key to looking at processes like when the protein changes its conformation, like what I showed in the beginning. So here's an example of a two-dimensional NMR spectrum. That's another beauty of NMR. You can do multi-dimensional spectroscopy, as many dimensions as you like, almost. So what we show here is just 2 dimensions. It's usually easier to look at 2 dimensions than 5. So here we have on the horizontal axis is the hydrogen frequencies, and the vertical is nitrogen frequencies. And each peak here represents one backbone amide, just like I showed on the previous slide, for one particular residue in the protein. So each of these, we can then work out that this peak belongs to that residue, etc. It takes a little work to, we have to run a bunch of different NMR experiments, but we can tell which one belongs to which here. So this is basically a fingerprint of the protein on that an NMR spectroscopist sees. And by applying different types of NMR experiments, we can encode these peaks, their shapes and heights. We can encode in those information that we want to extract, like the dynamics. So if we cut through, take a one-dimensional slice through one of these peaks, it may look something like this. This is a one-dimensional spectrum. And this slide here also introduces the concept of conformational exchange. So if we have our protein and it can exist in these two conformations, A and B, the spectrum will then usually have two different peaks, one from conformation B here and one from conformation A here. And if we have equal amounts of these, so populations are 50 and 50, then they are of equal height in blue here and here. A little bit hard to see under the magenta. And if they're skewed 10 to 90, then there will be one small peak, obviously, and one large peak. So this is if there are just two different species present. Now, if we say that these can actually interchange, so this conformation exists for a certain time, and then spontaneously, with some characteristic time constant, it moves over and becomes A and goes back and forth. If we, so to speak, turn on this exchange, the spectrum will change its appearance. So if this going back and forth is much bigger than the frequency difference, so go here from omega A, frequency we call omega in NMR oftentimes, and omega B, that's 200 hertz, is the difference between them. So if the rate going back and forth is much greater than that, then the NMR experiment, the spectrometer, can only see the average. It kind of, hope that's intuitive to you. And then what you see is the population weighted average of the shift. So for the 10 to 90 case, you have something that's very close to minus 100, minus 90 almost. And then if it's equal, it's right in the middle at 0 in this case. If it's much slower than 200, or the difference here, then you get a broadening of the peaks. Well, here it's broadened too, to some extent. But it's broadened, and the smaller peak can totally disappear sometimes. So you basically can't see it in the spectrum. Now, when we do these measurements, like I said, we can encode information by the specific experiment we run. We can encode information in the peak shape. So we can make the peak shape become greater or smaller depending on the rate that we pulse in comparison to the rate of this exchange. Then we can actually extract out all of this information. We can extract out the difference in chemical shift, and the chemical shift here you recognize has something to do with structure. So we get some structural information, and sometimes, which I will show later, we can get quite accurate structural information from these values. Although commonly they're hard to interpret. We get the exchange rates of the backwards and forwards rates, the sum of those, and we get the populations here. So we get structure, kinetics or dynamics, and we get thermodynamics. And that's basically anything that, or everything that a physical chemist is interested in knowing. If we know this, we know everything there is to know about a protein. Right, so back to the question, does it bind this way or that way, and how can we approach this? So what we did, or rather my students in the group did, they ran a bunch of these experiments. So now each of these dots here correspond to a line width, although it's measured not as a line width in a one-dimensional spectrum, but in a different way that I won't bore you with at this stage. So they measure this for the protein in the absence of any ligands, and then also at 7 different concentrations of the ligand present. So here the protein is saturated to 4%, and then you go up to 75% saturation. And you see that the shape of these curves change as we increase the ligand concentration. And we don't go beyond 75 because you see here it tends to get totally boring. It's a flat line, so that's not worth measuring at higher levels. And we can use all of this data and we can actually tease out all these different rate constants and get a lot of information, really a lot of information about what's happening when the protein binds the ligand. But first, let's look at what happened when my student Carl, when he measured only the protein itself. So he did these measurements, and you see that there are these curvatures in the lines. The numbers here correspond to different magnetic field strengths that we use in the experiment. Nothing much to bother with at this stage. And analyzing all the data, he showed that residues right here in the binding site, they show conformational differences, even in the absence of ligand. And he could measure the rate of this exchange, so going from state 1 to 2 happens 34 times per second, and the population of the alternative state, state 2, is only 4%. But we can easily measure this. And this is a side view of the same thing. Here's the binding site. And what about the chemical shift differences? These are just the global values that are common to all of the residues, because either the protein is in state 1 or in state 2, and when it goes between these, the entire protein senses the same thing, but it's only reported on those that have a chemical shift difference, the red ones. But the chemical shift difference itself also tells you a lot. It carries the information about the structure, remember? So in this case, we were thinking, how can we use these shift changes? Can we get more information out from these? And yes, you can. So what we do here is we plot on the vertical axis is the chemical shift difference that we get out from the experiment. And we plot that versus the chemical shift difference that you measure in the two-dimensional spectrum from the protein without ligand present, and when you have totally saturated the protein with the ligand lactose. And you see that these values correlate quite beautifully. They fall in a straight line with some error and some error bars. So what does this tell us? It tells us that the chemical shifts of the higher energy state, the state that only has 4% population, must have chemical shifts that are very similar to the lactose-bound state. And in turn, what does that mean? It must mean that this high energy state looks very similar, has the same structure as the state when the protein has bound the ligand. And you may wonder, why would that be? And we were lucky because we also determined the X-ray crystal structures of this protein. So shown here in blue is the trace of the binding site of the apoprotein, the protein without ligand. And in green, you see the protein when it has bound lactose. And you can see that these stay perfectly superimposed. They look exactly the same. For the green, we also have lactose present. In green, again, with red being the oxygen atoms in lactose. And then we have these pink spheres, and they correspond to water molecules that are present in the structure, in the blue structure, without any lactose present. So the protein actually binds water oxygens at exactly the same places as the lactose oxygens. So it has evolved to be a cradle for binding sugar-like molecules, or just a collection of oxygens that have the same arrangement as the oxygens in sugar. And it means that what we see in NMR is that once in a while, the protein goes from its ground state and populates a high energy state where all of these side chains come in and create the coordination pattern that coordinate water molecules in these positions. And this has a very, very short lifetime. It's only there for like a millisecond, and then the protein relaxes back down to its ground state. And you may ask, why on earth does the crystal structure look like the high energy state? And the answer is we don't know, but it could be because of crystal packing forces in the crystal between protein molecules that this one gets selected when the protein crystallizes. But anyway, we have a very high resolution structure of the high energy state that we see by NMR in solution. No crystal, just a protein swimming around in solution. So with this, we've actually measured one side of this quadrant or rectangle that we represent states 1, 2, 3, 4, right? This is the side. And this is the same. If we now add ligands, these kinetics won't change. They will be the same. So we have already solved part of the puzzle of measuring all of these rates that we're interested in. So next step then is that Carl, my student, he measured all of these, and he could then fit all of these data together, a model that includes everything. And then we can get, well, first I should tell you that what you see when you add lactose is that it's basically the same residues that we saw without lactose added that light up, that have a chemical shift change when it goes between states, free and bound, and also a few more because lactose now contacts other regions of the protein and makes it so that the chemical shift, the environment and the structure of its part of the protein changes. And then we get the rates at each of the different concentrations here. And all of this is summarized here. We get the rates of going from state 1 to 2, 2 to 1, the on rate of ligand coming into the binding site, the off rates from state 3 and the off rate from state 4, and then the backwards and forward rates when the ligand has already touched the protein, so to speak. Let's see if we have, and yeah, maybe we'll work from here, yeah. So what does all of this tells us? It tells us quite a bit. So here's just one example. We find out from this that the ligand affinity is much higher for the state that looks like the bound state than the state that can't bind the ligand. It's a factor of 270. So this then says that, aha, this is exactly what you expect if the protein binds the ligand by conformational selection. That is the upper right pathway. But it turns out that if we actually calculate from these rates the flux, that is the mass transfer flow through each of the arms, either the induced fit or the conformational selection pathways, the induced fit pathway totally dominates, and that's shown here. So conformational selection is this in blue, induced fit is in red, and in this red box here we've expanded that on the right-hand side. And you can see that in the absence of any ligand, and with very small amounts of ligand, conformational selection dominates. But as soon as you only have like 5% saturated protein, 80% of the binding goes through the lower left pathway, the induced fit pathway. And the reason for this is that the rate of going from the closed to the open conformation is about 1,000-fold faster when the ligand is present than when the ligand is not present. So this is basically amounts to a transition state stabilization of the protein conformational change. So this is in red is the case where you don't have any ligand present. You have a high energy state. This is the one that we think binds waters in a pattern that looks like lactose. There's 4% of this guy, and then you have a high barrier, so it's a slow process going from state 1 to state 2. But then when the ligand sits on the binding site without being actually in the binding site, when it's close there, it actually lowers the transition barrier. So going here is now requires much less energy. And then you have the bound state here. So this, I think many of you have heard about how enzymes work, how they stabilize the transition state. They lower the energy barrier, and that's how they speed up a reaction. And what we see here is that well, let me take one step back. So obviously the enzyme then will take substrate and turn it into something else. Some enzymes actually just change the conformation of the substrate. And here we have the reciprocal case where the ligand performs transition state stabilization of the reaction that is now the protein changing its conformational change. So this is something that's totally obvious from a physical chemical perspective. You would expect to this be the case, but it's probably something that is not very widely recognized in the biochemical field. And this kind of more widened, deeper view of ligand binding is not in the textbooks for sure. And it now opens the possibility to monitor these processes in detail, and maybe actually in the end it can lead to drugs that have enhanced performance in various ways. So this was a research talk of sorts, and I want to thank the students. Olof, Carl, and Christopher is my colleague who worked on this. I should say that this has been a very, very long-term project. Olof here, Carl, he started this project 15 years ago. So he's no longer in the group, obviously. And then Olof took over. Back in 15 years ago, we didn't really understand what was going on. And then Olof took over, and we knew how we should approach it. And he collected a lot of data. And then he had to redo it twice. And then finally we had made sense of everything, and we could trust the results, and we could publish the data. And I also want to thank some nice people for money. And most of all, I thank you all for listening. Thank you. Are we taking questions?